Hey, welcome to those online and in person. And this is truly the most people I've seen in this room in many years. So I think this is exciting. So for those who don't know me, my name is Andrew Goldstein. I'm a professor of molecular cell and developmental biology, and I'm thrilled to introduce our speaker, Dr. Costas Lyssiotis. He's a professor of molecular and integrative physiology from the University of Michigan and a true leader in metabolism research. I invited him a few years ago. He had a sports injury, so I'm glad he was able to make it now. And so Costas was an undergrad at Michigan, did grad school in San Diego at Scripps, a postdoc in Switzerland. Is that right? Briefly. And then moved to Boston to work with Lou Cantley, and then moved to Cornell with him before being recruited back to University of Michigan, where he has quickly risen through the ranks. He has the remarkable number of publications, collaborations, and has just made really impressive, substantial contributions to the understanding of normal physiology, cancer biology, and most notably cancer metabolism, focusing not just on tumor metabolism, but also on the microenvironment, the immune cells. His work in pancreatic cancer is really leading the field. He's earned a crazy number of awards from the American Cancer Society, V Scholar, Young Investigator from Melanoma Research Alliance, Sidney Kimmel Foundation Scholar, and is one of the most highly cited researchers in the field. I'm absolutely thrilled to hear his science, and I can't wait to learn. So welcome, Costas.
All right. Well, thanks, Andrew, for the really kind introduction and the invitation and your patience. You invited me in, I don't know, 21, and here we are. I'm not going to go through all the gory details about my recovery from a soccer injury. It's not interesting. I do want to touch on one point, though. So for the graduate students in the lab, I did a 3-month postdoctoral fellowship. It was part of a short-term fellowship program in between my PhD and my formal postdoc. And I got to go travel to Switzerland. I ended up working with Doug Hanahan. Those who work in cancer biology know who Doug is. It was a really awesome opportunity. It was something that I hadn't heard of until another postdoc came into our group and told me about it. So I do, when given the opportunity, like to bring it up, particularly for the graduate students. I think a great learning experience. It turned out to be a lot of fun as well. Okay, huge digression there. So today, we're debuting a new story here, targeting lipid metabolism in pancreatic cancer. I'd love to hear feedback on this story. We have it out, it's pre-printed, and I'll show you that link and hit on a couple of high notes before I get into the story, but very eager to get feedback on this, on its reception. I was going to talk about this story today, and I changed at the last minute because this may resonate with maybe half of the room. Brandon just came through UCLA and interviewed with, I think, just about every member of the faculty. And I'm like, Brandon, were you just at UCLA giving this talk? He says, yeah. I said, well, I better switch gears here because everyone's going to leave. They're going to eat their nachos and take off. But so if you didn't hear about Brandon's story, he's got, Brandon is a graduate student with Yatrick and me, and he made a really cool discovery about how BRD4 regulates interaction between mitochondria and endoplasmic reticulum, and what that means for cellular physiology. This is on bioRxiv. We're dealing with the revisions now. It's a really exciting story, but you all know that because you already heard about it. Okay, so what I'm going to talk about today is pancreatic cancer. I don't have a good sense for who's in the audience, what disciplines y'all come from. So I'm going to really start from the top and then we'll narrow it in. If you're not a cancer biologist and you haven't heard, maybe it'll come as a surprise. If you are a cancer biologist, this is all the rage now. We appreciate that tumors are not just masses of malignant cells, but rather a better way to think about a tumor is as a pseudo organ. So a cell acquires a mutation and it reverts its differentiation state into more proliferative state. And in the process of establishing a tumor, it is enacting some aspects or reenacting some aspects of its development, which means that it calls a bunch of other cell types in and tries to to recreate its developmental origins and recreate an organ. So that results in a number of cell types that interact with the cancer cells during the establishment and progression of cancer. What we've come to appreciate in the field of metabolism is that across all solid tumor types, what we observe is disrupted metabolic features. So this is typically manifest by leaky and insufficient vascularization of this pseudo organ. And this is experienced by the cancer cells in other cells in the tumor microenvironment in a number of ways, not the least of which are varying access to things like nutrients and oxygen. Certain aspects of the tumor will accumulate metabolic wastes, changes in pH, and so forth. So the big question my group has been interested in understanding is how does a tumor manage these metabolic changes, these metabolic alterations, and we know that the tumors are growing, or it wouldn't be something that we talk about. How do they even harness some of these aspects of deregulated metabolic physiology to their benefit? So we study this, my group studies this in pancreatic cancer. What I've pictured here is a representative histological image of a pancreatic tumor. And you don't have to be a histopathologist to appreciate that. Because I've done a little bit of the work for you, I've circled the neoplastic epithelial cells, that most of this area is not in fact cancer. Most of this area is other stuff. So what is all that other stuff? In pancreatic cancer, upwards of 90% of the tumor can consist of this other stuff. For pancreatic cancer, most of it is fibroblasts, or as I will call them a little bit in this talk, CAFs, cancer-associated fibroblasts. And there's a lot of immune infiltration as well. In pancreatic cancer, most of that is myeloid-derived, and we call these tumor-associated macrophages. The tumor microenvironment imposes a number of unique features on pancreatic tumors, one of which, if you are a histopathologist, you'll appreciate that there's very little vasculature in pancreatic tumors, making them, in fact, the prototype for avascular tumors and really setting the stage as a great example of how are these cells growing if they don't have access to blood-derived nutrients. And this brings up some of the major questions in the group. How is metabolism in the cancer cells or in the tumor rewired to support the growth and survival of the tumor, its therapeutic resistance? You know what, I forgot to mention one more point here. Just give you a 10,000-foot view of the clinical characteristics of pancreatic cancer. This is the disease. It's the worst major cancer. And a big part of that is late-stage diagnosis, but another big part of that is our therapies don't work. So the 5-year survival of a patient diagnosed with late-stage disease is around 2 or 3%. These numbers are crazy, 2 or 3%. We have the same medicines working in other cancer. Why aren't they working in pancreatic cancer? So, and immunotherapy doesn't work in pancreatic cancer. So these are some of the questions that have motivated our group to focus in on aspects of the tumor microenvironment and aspects of metabolism in order to better understand and ultimately target these. So how is metabolism, getting back now to the slide here, how is metabolism rewired to support this therapeutic resistance? We know these tumors are growing. Where are they getting the nutrients? Right? Where are they purposing those nutrients from? That's going to become central to today's talk. This is another area I'm not going to get into nearly as much, but that we're interested cooperative metabolic pathways among different cell types in the tumor. I'll tell you more about that in 1 second. And then, do the deregulated nutrient profiles influence the way that the immune system senses, responds, and is able to take down the tumors? The anti-tumor immune system, like the cancer cells, requires nutrients to do its functions. Does the lack of nutrients impose barriers to immune recognition? And as we're thinking about all of these questions, we want to know will these lead to any new inroads to develop new drug targets and therapies? And the story I'm going to tell you about today, I've been working in cancer metabolism now for about 15 years. This is the most exciting target and response I've seen across all the models we've looked at. We're moving this into phase 2 clinical trials now. Those stories are ongoing, but that's just to whet your appetite. I'll tell you more about it in a second. All right, so coming back to this question, pancreatic cancer cells, pancreatic tumors have limited nutrient environments. Where are they getting the nutrients they need? And is this imposing other aspects on the pancreatic cancer biology? What we've come to know as a field, my lab's has contributed to this work, but this is a large area of research by a number of labs. And the punchline has been that pancreatic cancer cells, in order to manage not having access to blood-derived nutrients, they become expert scavengers and expert recyclers. All right, in a punchline. So let me take you through 2 examples of that. This is an area that we've been very excited in. This is metabolic communication among cell types. So I would call this this is more of the scavenging arm. We know that different cell types in a tumor will communicate by way of metabolism by cooperatively sharing nutrients. Maybe a waste from this becomes a fuel for this, and they create these crosstalk pathways that are utilized during other aspects of normal physiology and organ growth, but that are repurposed by the cancer cells. So that's cooperative metabolic crosstalk pathways. There are competitive metabolic crosstalk pathways. These are largely targeted at the anti-tumor immune system, as I alluded to on my last slide. In the case of these pathways, the tumor, sometimes the cancer cell, sometimes other cell types in the tumor microenvironment will gobble up all of the nutrients, not necessarily for their own purposes, but to keep them away from the anti-tumor immune cells. This has now emerged as a major tumor suppressive mechanism by which tumors will keep the immune system at bay, and we've had some success targeting these pathways. They don't act necessarily directly on cells in the tumor, but what they do is free up the nutrients that the T cells can now use to attack cancer cells. We've also done some work to illustrate that metabolites that are available in the tumor microenvironment act directly as mediators of therapeutic resistance. These are not mutually exclusive, but we just think this is a useful framework to think about the scavenging. I just want to put that in there. It's a little bit of background perspective, but what I'm going to talk about today is this idea of recycling. And what we've come to appreciate is that pancreatic tumors and the cancer cells themselves do an excellent job making use of the nutrients that are available inside of the cells and in the microenvironment that are processed through the lysosome and provided to the cell. I'm going to dive into that a little bit deeper because this talk is going to center around autophagy. So in around 2010, Alec Kimmelman's group made the fortuitous observation that pancreatic tumors have elevated levels of autophagy. His group, Eileen White's group, a couple other labs have subsequently gone on to demonstrate that this elevated autophagy leads to the catabolism of a variety of different substrates in the lysosome. Right around the same time, so this was about 2010, in around 2013, initially Cosimo, then in Daphne Barsagi's lab, and subsequently a number of other labs, illustrated that in addition to using the lysosome to break down intracellular cargo, that pancreatic cancer cells in a RAS-dependent manner will non-specifically engulf extracellular contents and similarly break those down in the lysosome, and all of the goodies that come out of these 2 pathways can then be used for various purposes. How the cells turn on lysosome biogenesis was demonstrated by Rashika Pereira when she was in Nabil Bardizi's lab, and she illustrated a transcriptional mechanism by which this pathway is hyperactivated in cancer cells, This gives us some idea about the therapeutic window, why these cells will, as I'll show you, become selectively dependent on autophagy. A major question that emerged from this collective body of work was, what is the point? Why are these cancer cells becoming dependent on autophagy? So we have a sense for how it gets turned on. We know that it's turned on. What are they using it for? So Alec and Eileen White's work demonstrated that this was, at least in part, to generate nutrients that could be used to support cellular biosynthesis and bioenergetics. Alec and a number of other groups later showed that this was also a major mechanism by which iron in the cell bound to ferritin gets turned over so that the cell can use iron for a variety of purposes. One of the more provocative studies released recently by Alec and Rashika's labs is that autophagy is also a mechanism that pancreatic cancer cells use to take MHC1 off the cell surface. And so they become less reactive to T cells because they're displaying less antigen. And this is occurring in autophagy by way of autophagy, so selective autophagy. This has captured the attention. I mean, this collective body of work, it's extremely robust. A number of labs have contributed. This is well recognized as being one of the therapeutic liabilities of pancreatic cancer cells. From a metabolism standpoint, we've all been eagerly watching the trials unfold. And those trials are largely centered around using these agents, chloroquine and hydroxychloroquine. There are other chemical tool compounds. The chloroquine, hydroxychloroquine drugs have some clinical utility in non-cancer, but in pancreatic cancer, they have completely failed in all the clinical trials examined. And the consensus has been because they are just not great drugs. So when you look at biomarkers of response, they don't appear to be engaging target at the concentrations that we can deliver them in vivo. Other groups have also been looking at mechanisms of autophagy resistance. And so if you inhibit autophagy, how does the cell respond to that? And so that's going to become a major focal point of today's talk as well. That's work that was led by Kirsten Bryant in Channing Durr's lab. Okay, so I hope at this point I've convinced you of a couple things. Metabolism is deregulated in pancreatic cancer. They manage it at least in part by activating autophagy. Autophagy is a provocative drug target, but the drugs suck. Okay, so if you've been sleeping, that's the punchline up to this point. Maybe I gave you too much detail. I thought I'm sort of debuting this talk. I'll pick on you guys at dinner tonight if that was too much information. So how do we target autophagy? We think we have a new inroad, and that is using this drug called ESK-981, and I'm going to tell you a lot about that today. ESK-981 is an inhibitor of PIK5. PIK5 is a lipid kinase that phosphorylates PI3P on the 5 position to make PI3-5P2. I'll talk, if I refer to that, I'll probably call it ESK. There's another tool compound that exists called apilimod. This one has been in the literature for quite some time. It's a great PIK5 inhibitor, but it's a terrible medicine as well. But we've interchangeably used these for a number of our assays because they're independent pharmacophores. And so they give us confidence that, you know, if you look at the Venn diagram, the thing that they both do is hit PIK5, but they both have off targets. We use genetic approaches too, and I'll cover that. So how is PIK5 related to autophagy? Well, the formation of PI3-5P2 is a marker that brings late endosomes and autophagosomes into contact with the lysosome. And so blocking this phosphorylation event blocks the trafficking of these organelles with the lysosome and therefore inhibits autophagy. That is not the only thing it does, as indicated by the late endosome, but one of the things it does is to inhibit autophagy. That's the collective background summary. That's the collective background. And so this is the story I'm going to tell you about. I'd like to take a quick break and give you a snapshot of the overview of the talk at this point. I think it's a helpful way to follow along. This also sets the stage so that I can remember to acknowledge the outstanding investigators that did all of the work here, and also gives me an opportunity to point out that we recently put this manuscript on bioRxiv. So if you want to know more and you don't hear, if you have more questions or you want to know more details, I definitely encourage you to check out the bioRxiv. We pre-print everything. And that's another discussion we can have if time permits or whenever. All right, so this work was led by an extremely talented MSTP student at the University of Michigan, Caleb Chang. Caleb is jointly mentored by Rul Chanayan and me. And Yuan Chao is a senior scientist who, as I'll show you, really got this project kicked off. So under normal circumstances in pancreatic cancer, that's what PDAC is, pancreatic ductal adenocarcinoma. Sorry for the acronyms here. Metabolic homeostasis, at least as it relates to today's talk, is regulated by this MAP kinase signaling pathway downstream of KRAS to generate lipids de novo. And the other way that lipids are generated or provided to the cell is through autophagic turnover, which is mediated by PIK5. When we inhibit PIK5, that blocks this part of the pathway, and the cells become more dependent on RAS MAP kinase signaling to make lipids de novo. Reciprocally, if we inhibit RAS or MAP kinase signaling, the cells become more dependent on autophagic turnover. If you haven't been following along, there are some really great KRAS inhibitors that are making their way into oncology, into clinical trials. And so there's been a lot of excitement and attention here. 95% of pancreatic cancers exhibit a mutation in KRAS. So this has been a really exciting area for the field. So the fortuitous opportunity to combine it with autophagy inhibition, that's one of the things that's got me really excited. And further, once you see the responses we're seeing in our preclinical models, I trust that you'll feel similarly. Anyhow, what these two demonstrated is that if you inhibit either one of these two pathways, you create a synthetic lethal context in which, when both pathways are inhibited, the cells really suffer. In fact, they die when you inhibit both through apoptosis. We've seen tumors completely melt. We've not observed major toxicities in the animals for reasons that I'm still trying to wrap my head around, but that's the biology. And so that's the punchline of the story. That I think this is, we are moving into clinical trials with this. And like I said, this is the most exciting observation I've seen in pancreatic cancer during my time in the field. And this is a disease for which we really need new therapies. Okay, so I'm done. I'll see. Just kidding. That's a summary. Now let's get into some of the data. That was the first time I debuted that joke. Let's see. Didn't go over that well. 1226. We're not doing bad. All right. So where the hell did PIK5 come from? Or this PIK5 inhibitor ESK come from? This is work that was led by Yuan, and she's in a rules lab. She published this paper in Nature Cancer in 2021. And what she had, I'm just going to cut right to the chase here. The experiment that she started with was to take a panel of kinase inhibitors that are clinically approved and treat neuroendocrine prostate cancer. Completely different disease. But this is another challenging disease. I don't know if anyone studies prostate cancer. Don't ask me any questions about it, please. But what she found is that there was this one compound that really stood out. And then when she went after its, when she, I guess, just compared its targets to other compounds in this library, she saw that those other targets were well represented. However, they were not exhibiting the same activity. So she started digging into what this activity could be, and she deorphanized ESK-981 as a PIK5 inhibitor. It still has polypharmacology, as most drugs do. And so that's why we had to use a number of orthogonal approaches to illustrate the effects we're observing in pancreatic cancer, and that the Chanayan lab has observed in neuroendocrine prostate cancer is through its inhibition of PIK5 and inhibition of autophagy. So Rules Group was excited about this for neuroendocrine prostate cancer, and they had completed a phase 1 trial. So this medicine is safe. It's now in a phase 2 trial recruiting for not just pancreatic, but for a number of different types of cancer. And around this time, Caleb joined my lab and a Rules lab, and a Rules that I've heard that autophagy is important for pancreatic cancer. Let's see what's going on here with this medicine, because we're seeing really cool responses in our neuroendocrine prostate models. So here, if you look at our cell lines, this is ESK IC50 shown on the y-axis. At your left are a number of prostate cancer cell lines. They are highly responsive. These two are neuroendocrine. These are non-neuroendocrine. If you look at our pancreatic cancer cell lines, they're even more responsive. So we got excited. Maybe there's really some traction here. Couple dose response curves using different compounds or different pharmacophores to illustrate that all of these pancreatic cancer cell lines are responding to PIK5 inhibition, at least presumably at this point in time. The next thing we did was, Caleb and Yuan did, was to take this compound into a couple of our animal models. I'm going to show you phenotype, and then I'm going to go back to mechanism afterward. What they found was, as a single agent with this drug, ESK, that they were seeing some pretty remarkable responses. This is better than many of the other metabolic inhibitors we've tried in this disease. This is, again, single agent. No real cures here, but there was activity. This is a murine syngeneic allograft model, which means these are immune competent. This is a human cell line derived xenograft model. You can see pretty good responses as a single agent. If we take another human cell line derived xenograft model and we look at tunnel staining to mark apoptosis, you can see that in the ESK treated samples, we are inducing apoptosis. That is also revealed here by looking at PARP cleavage. So we know that we're able, not just inhibiting the growth, but we're actually killing cells. Again, this is single agent. We also know that, well, if you look at this model, for example, these tumors will grow through single agent. So this is not a silver bullet. This isn't going to work by itself. But we believe that at this point in time, we had a reasonable handle to start digging a little deeper into the biology. So we made the conditional knockout model. We crossed that with a pancreas-specific CRE. If you knock PIK5 out of the pancreas, here's some histological images of beta cells, of islets. You can see some acinar tissue around there. I'm not going to go through this at length. It is all in the supplemental data. The pancreata are completely fine when you knock PIK5 out. If we then cross that model with a mutant KRAS allele, so I told you mutant KRAS is the most prominent and signature transforming oncogene in pancreatic cancer. This is the model we use for our genetically engineered mouse models. We put KRAS into the pancreas. These animals will get disease, as you can see, that initially is early transformation, ultimately through a loss of a tumor suppressor. Those animals will die of primary disease burden around 60 weeks. If we have either homozygous or heterozygous knockout of PIK5 on that background, you can see that these animals live for much longer. What really caught my attention here is that we are seeing similar results with the heterozygous animal and the homozygous animal. I still don't know what to do with that, but I think from a drug development perspective, which is not something that my lab has ever claimed to be good at, but it feels like that's a good sign. You don't need to inhibit the activity 100%. You can do some blockade of the activity, which is what a drug does, and still see a pretty remarkable response. All right, so now let's get a little bit into the biology and into the mechanism. And at the end, I'll take you into what I consider to be the really exciting aspects about synthetic lethality relating to RAS pathway inhibition and PIK5 inhibition. So if we take the PIK5 inhibitors proposed to be autophagy inhibitors based upon Yuan's earlier work and the known activity of PIK5, what we can see is that if we put either ESK or a pilimod onto these cells, you can see relative to the vehicle-treated cells, the vacuolization occurs. That is, that's a readout for inhibition of autophagy. You can see that's the same is true if you use a genetic approach. Caleb is a very thorough scientist, and he did a number of assays to illustrate that ESK was indeed inhibiting autophagy through PIK5 inhibition. Here's a number of other markers of autophagy inhibition. I'm not going to take you through all of these because I don't want to bore most of the audience. But those of you who are really interested in autophagy, you can check out the preprint. We also used a reporter model where we can look at autophagy index if you use either apilimod, ESK, or chloroquine. This is a lysosome inhibitor. You block autophagy. If you induce autophagy by inhibiting mTOR, so when mTOR is inhibited, autophagy is induced. It's a well-established positive control. Even in that context where you hyperactivate autophagy, you're able to bring it down using our 2 PIK5 inhibitors plus the other positive control chloroquine. So we know that at a minimum, there might be other impacts of ESK on cellular physiology, but we know at a minimum it is inhibiting autophagy. So to get a sense for what it's doing in these cancer cells and why it has such a potent response, again, even as a single agent, and in our cell culture models, it's even more effective probably because we can dose it harder. Caleb he took 2 parallel approaches. So the first was to perform RNA-seq. And what he saw with RNA-seq is that we activated a pro-lipogenic gene expression program. Some of the key genes are indicated with red dots here. This is a gene expression pathway analysis. You can see there's activation of fatty acid and cholesterol metabolism, among other things. We also saw that when we treated with our PIK5 inhibitor, we were inducing a pro-lipogenic gene expression program, and this was occurring downstream of SREBP. So I don't know if Peter's in the lab. I love, maybe he's online. Oh, students from Peter's lab are in the room. I'd love to get some more feedback on the thoughts around SREBP. We haven't dug too deep into this, but it is clearly this lipogenic gene expression program is occurring downstream of SREBP. We also looked through lipidomic profiling that there was, so what I have, let me walk you through this relatively complicated diagram. Lipid class is on the y-axis. Change in the abundance of these varying lipid classes is shown as log2 fold change on the x-axis. Blue is the apilimod, and red is ESK. There's just a remarkable convergence of these 2 independent pharmacophores on the same aspect of lipid biology. What we're seeing is that a number of ceramides are being increased when we treat with a PIK5 inhibitor. Also, I will tell you, we're not really clear on what this means either, but we took this collective information and we harmonized it with the data that came out of a CRISPR screen that Caleb ran. I trust everyone's relatively aware of how CRISPR screens work, but in essence, we took a metabolome-targeted CRISPR library, we treated cells with that library, plus or minus the PIK5 inhibitor, and looked for genes that were either selectively enriched or selectively depleted. The depleted genes would suggest that those may be targets that we could use combinatorially with PIK5 inhibition, and the enriched genes would give us information on mechanistically how the cell may be adapting. Okay, so for today's talk, I'm just going to focus here on the bottom, those genes which were selectively depleted. If you are an aficionado in this space, I imagine some of those genes are going to pop right out at you, probably if you're in Peter's group. And if we map those genes now onto de novo lipid biosynthesis, what you see are among our top 10 hits were the citrate exporter and ACC1 and FASN, which are involved in de novo lipid biosynthesis. All right. We also saw a number of genes involved in elongation and in ceramide biosynthesis. But for the purposes of the subsequent slides, I'm just going to draw your attention to these two rate limiting steps in de novo fatty acid biosynthesis. We also noticed cholesterol metabolism come out of our RNA-seq, but as you can see up here, cholesterol metabolism did not score on our CRISPR screen. So we think that that was more likely a consequence of being downstream of SREBPs than being functionally relevant to the activity of PIK5. All right, so what Caleb then did was, all right, maybe we have these handles now where we can inhibit PIK5, come in with another target, and operate on the synthetic lethal axis. So what Caleb did was to knock out FASN, either, let's see here, individually in red, or combinatorially with apilimod. This is one of the PIK5 inhibitors I've been telling you about. And you can see that individually, modest effects, together the effect is more substantial, though we're not killing cells when we do this. This is with CRISPR-Cas. He then did the same with an inhibitor of ACC1. That's illustrated here on this Western blot by a block of phosphorylation. And you can see that individually, again, modest effects, but combinatorially, we had a much more pronounced effect. The cells still appeared to be growing through this. So this didn't seem like it was going to necessarily provide us with the therapeutic approach that we were targeting, though it was interesting mechanistically, and I think it led us on the right track. So we decided to take a step back and ask, how were FASN and ACC being regulated in pancreatic cancer? And my lab has a whole history of publications about how mutant RAS regulates metabolism in pancreatic cancer. And because mutant RAS is such a prevalent oncogene in this disease, it also seemed like the most logical place to start. So that's what Caleb did. This is a really a lot of data, but I'll walk you through it briefly. The punchline is straightforward. KRAS MAP kinase signaling activates de novo fatty acid biosynthesis. So here we have, these panels are a mouse line. These panels are a human cell line. Here we're using a KRAS G12D specific inhibitor. Here we're using a MEK inhibitor. So that's going to block MAP kinase signaling. This is a G12C inhibitor. This is a MEK inhibitor. If you apply those to these pancreatic cancer cells, we bring down FASN and ACC1 expression at the transcript level. That's also represented here at the protein level. It's a lot of information. I don't expect you to take it all in. It is in the bioarchive, but just hitting the high notes. This is another model in which we can remove RAS signaling by taking doxycycline out of the system. I won't go through all the details there, but just know that we can turn RAS off by pulling doxycycline. So this is a third independent model where we illustrate that when you remove RAS, FASN and ACC expression are decreased. So in essence, to make a long story short, that it was these data that brought Caleb and Yuan to their central hypothesis, which was, it seems like when you inhibit PIK5, RAS takes over. It seems like when you inhibit RAS, PIK5 takes over. And targeting either ACC or FASN isn't doing the trick. Let's hit a little higher on RAS, and let's do them together and see what happens. So the first experiment that they did was to use a human patient-derived xenograft model. So this is UM19. This is one of the cell lines that came out of a clinic working with the pancreas team at Michigan. You can see here with trametinib in red, that's the MEK inhibitor, single agent, very little effect. ESK has a modest effect alone, but obviously the tumors just grow right through it. The combination completely eliminated these tumors, completely eliminated. The animals went on to be totally disease-free. I had not seen a lot of experiments like this, but there are a number that do exist in the literature, just not from my lab, using this model system. Okay, so this is maybe a lower bar to jump over. So we then wanted to move on to more physiologically accurate model systems. And here we turned to model system in which we put the pancreatic cancer cells into the pancreas. That's an orthotopic injection. And we did so using a syngeneic cell line so that we could have an immune system present. This model system, Caleb and Yuan did in essence the same thing. They treated with ESK plus or minus trametinib. And it's probably best represented here. And I'll tell you what the bar graphs mean just in 1 second. Independently, if you look at CK19 as a tumor-specific marker, there's a whole bunch of tumor. Individual treatment in this model slows down the tumor, but you can still see that there's a lot of tumor present. The combination, these animals are completely tumor-free. That's illustrated here in these bar graphs. So on the y-axis, this is pancreas weight plus tumor weight, vehicle, mock treatment, individual arms, and the combination. This residual weight is because there's a pancreas. And so I told Caleb and Yuan, I like, this is such an amazing result. You need to show what a normal pancreas looks like so that people can see that the pancreas does carry its own weight, and that's the case here. If you quantitate this area, that's illustrated here. There's no pancreatic cancer in these mice. We've taken these mice out now. We took them off therapy and let them, live normal, healthy mice lives. We're several months out. These mice are still tumor-free. We've repeated these studies with a KRAS specific inhibitor, in addition to this MEK inhibitor. Those mice are still going strong as well. This is, I would say, the second highest bar one can hurdle when trying to treat this disease. There is a even more difficult preclinical model. And in fact, it's so difficult that I'm not aware of anyone who has ever cured that mouse. This is the genetically engineered autochthonous model where it has RAS and P53 mutation. I'd love to show you that data. We don't have it. Those studies are ongoing right now. We're doing it with Jen Morton at the Beatson. Positive results from that would be, I guess, what we would need to move this no holds barred into the clinic. But we are still moving this into phase 2 clinical trials now, the PIK5 inhibitor, because it seems like it might have activity in neuroendocrine prostate cancer too. But I hope I was able to relay how excited I am about these results. I just showed you one of many phenotypes because it's kind of boring just to look at phenotype after phenotype. So you can go into the bioarchive if you'd like to see more model systems that we've tested. But let me bring you here to the conclusions. We got plenty of time for questions. What I hope I've relayed to you is that PIK5 inhibition disrupts autophagy, and that leads to activation of de novo fatty acid biosynthesis. It also, in doing so, creates a synthetic lethal opportunity where we can target RAS. And there's a ton of enthusiasm in oncology right now for RAS. We're using RAS specific inhibitors. We thought this was a target that was undruggable. Some really great scientists have illustrated that that's not the case. And we now have medicines that we can use to test these hypotheses in people. There's a lot of optimism around this. And then particularly when there's a therapeutic combination option, like ESK, which I told you at Michigan, we have in a phase 2 clinical trial. Oh, I mentioned this out of sequence, but there you go. Yeah, all right. So this is the group. I owe a special round of thanks. I've mentioned them throughout, but here again is Caleb and Yuan who did all of the hard work here. It's been such a pleasure working with both of them. This is a rules, this is not a small village in Michigan. This is a rules research team in numbers in the hundreds, but nevertheless, a rule is still very available and approachable. It's such a delight to work with him. Probably goes without saying, he has a lot of resources at his disposal. Other collaborators who've contributed, I didn't necessarily mention them by name going through, but we got a ton of help on the CRISPR screening from my good friend Yatrick. Marina is a pancreatic cancer expert and good friend. V is leading the clinical trial. Jen is doing the KPC study in the UK right now. Several others. thank you all for your attention. Thank you for the invitation. Thank you for the invitation, and I'd be delighted to take any questions.
Yeah.
And Lucy has this ball to throw around at you too.
Can I ask the first question since
Sure.
since I invited you?
Yeah,
So,
in there's prerogative.
so the kind of synergism between the lipid synthesis and autophagy, does that suggest that getting lipids is the main purpose of autophagy, or is it just some other feature?
I think that's a great question. Some of our data would seem to suggest that for sure, but I don't want to say that with certainty because there are so many other studies out there illustrating other roles of autophagy. And I also, even though I talked a lot about autophagy, we don't have data to specifically illustrate that the effect of ESK is uniquely on autophagy. There might be some other aspects of its polypharmacology or even its PIK5 specific activity that may be playing a role. So it seems like the activity downstream of PIK5 is for lipid biosynthesis, but not necessarily the effect of autophagy. I know I muddied the water there, but I did so somewhat intentionally
Sure.
because, I mean, it's science, it's dirty. I'd love for the models to be pretty and clean, but sometimes they're not quite that. Yeah, it's a great question. Thank you. Yes.
That was really.
Oh, I love it. I've never seen the ball in
No,
action.
They have these at Genalia and they make me real nervous because they throw them at people at Genalia and I'm real scared of it. This is really fantastic. I was wondering, especially with your work looking at like the autophagic flux and the inhibition of fatty acid biosynthesis, if you guys had ever like looked at the like if AMPK is playing a role in any of this or if you speculated on that at all.
We haven't looked. Yeah, we haven't looked in any meaningful way at AMP kinase, to be honest with you. There were a number of, that's what I'm trying to think, in our CRISPR screening library, components in AMPK signaling. For example, I think we see LKB1 in there, maybe some of the subunits of AMP kinase, and those didn't come out of the screen. So I think that's probably the best evidence we have, but otherwise we haven't looked at it. Good thought. Mm-hmm.
Can you hear me? Yeah, okay. Is it possible to buffer the nutrients for pancreatic cancer treatment? That is because the cancer, pancreatic cancer depends on nutrients. So are there nutrients that compete with the nutrients that increase the aggressiveness of the cancer cells rather than reduce the aggressiveness of the cancer nutrients?
Okay, I may not answer your question directly, but let me just riff for a second. So changing the diet for these animals and for people with pancreatic cancer doesn't do anything meaningful. So if you take away one nutrient, put them on a ketogenic diet, right? Take away carbohydrates, put them on a non-fat diet. We've done that in the lab, take away fats. That has no effect on the growth of the cancer. And I think we as a field have learned over the past 15, 20 years that cancer cell metabolism isn't highly adaptable. Unfortunately adaptable. It's a lesson we should have learned from signal transduction, but we had to learn the hard way. So since that seems to be a dead end, one of the approaches that our group has taken to sort of pivot around that is we know that the cancer cells are gobbling up nutrients. And they do so not always because they need those nutrients, but in order to keep them away from T cells. As an example, what we found is if you inhibit arginine catabolism in pancreatic cancer, so you leave more arginine behind, it's sort of the opposite of your question, that makes the T cells better able to recognize and to take down the tumor. It has no impact on the cancer cells when we remove their ability to catabolize arginine, which suggests to us that they are breaking down arginine as a tumor suppressive mechanism, not because they necessarily need those nutrients. Now, I give you the arginine example as a singular example. I think thematically that's going to apply to other nutrients, though not necessarily all nutrients.
What I mean, is it successful?
Say it again.
Is it successful?
In preclinical models, blocking arginine catabolism is successful. There are medicines now in clinical trials testing that concept. We're not involved with that work, but I think it's something we should be looking at. Yeah, thanks for that question. Got the ball.
Okay, so Could you explain again how the combination of ESK 981 and trametinib leads to sustained tumor regression? And are there any known like resistance mechanism that tumors might develop against this treatment combination?
Yes. I love the second question. So I'll just briefly show you, this is the model system again. So you said, how is the combination working? Most of the data seems to suggest that it's centering around, so this is sort of related to what Andrew said, and I gave him a squishier answer, but I'll give you the clean one. It seems like RAS signaling is activating de novo biosynthesis, and it seems like autophagy or some activity downstream of PIK5 is providing scavenged lipids. The activity of either one of these pathways is necessary for homeostasis, where if you block one, this one takes over. If you block the other, this one takes over. But if you block them both at the same time, again, for reasons that are not entirely clear why this is cancer specific, the cancer cells die. This is well tolerated in our animal models. So the animals have not exhibited any negative side effects. As far as mechanisms of resistance, I mean, the mechanisms of resistance emerge to everything we ever try. Cancer is enormously adaptable. That's what it does. So we are growing mice out. We are letting these mice age in which we've cured them of their primary disease burden and waiting for the tumors to come back. I'm all but certain they're going to. They haven't yet, but I'm all but certain they're going to. At which point in time, we're going to start studying mechanisms of resistance. So I love that idea. We don't have tissue available yet, but I think if it doesn't come out of our transplant models where we put cancer cells into the pancreas and let those, I'm sure it's going to come out of the work we're doing with Jen Morton on the autochthonous model. So that's a model that we engineer to express RAS mutant and P53 mutant in the pancreas. And those arise during the course of normal animal development. So those are the tumors that are real nasty. They look very much like human disease. We've never cured one of those animals. The field has never cured one of those animals. I imagine we're going to have resistance there. And I look forward to taking those tumors out of the mice, putting them on plastic, making cell lines, and beginning to understand how they're acquiring resistance. So yeah, I love that line of thought.
Hi, thank you for a great talk. You showed with your mice model lacking the immune system a much stronger or more profound response to either single treatment or combination treatment. Do you have any idea what the role of the immune system is under these treatment conditions?
Yeah, I love that question too. No, not yet. But I'll make 2 points because I'm long-winded and I have the microphone. So the first, there's this observation from Alec and Rashika that if you inhibit autophagy, the cancer cells express more MHC1 on the cell surface, which makes them more recognizable by the anti-tumor immune system. I imagine that's going to play a role. We don't have any evidence to suggest it does at this point in time, but if you inhibit autophagy, that's been demonstrated. And so that's one of the reasons I'm also enthusiastic about this combination, because it takes into account the immune system, which is something that metabolism people, at least for a long time, neglected. The second question, or my second point rather to your question, is the animals that don't have, that we've allowed to grow out and that are cancer-free, we're going to re-challenge them now. So in fact, I think Caleb set up that experiment today. We've been talking about it, where he's going to take tumors and put them in subcutaneously and see if they are rejected, if there's immune memory, in essence, from having rejected those primary tumors. Stay tuned. Yes.
Hey, this is a super cool work. So it's very clear that lipid homeostasis and de novo lipid synthesis is essential for pancreatic tumor growth. But so lipid metabolism involved in multiple cellular processes. For example, lipid can be substrates of beta oxidation to generate ATP to provide energy. Also, lipid can be the source of phospholipid to generate membrane structures. So I was wondering which aspect of lipid metabolism is essential like in this study, like as a mechanistic inhibiting the pancreatic tumor growth?
That's the $1,000,000 question. And I wish I knew more to tell you something smart and meaningful. We're starting, I mean, honestly, I'm a little out of my depth here. We are starting to delve into that pathway, and we're using as a handhold the data that came out of the CRISPR screen. So it looks like the production of sphingolipids and of various ceramides is important. And I think we now need to know, start asking the question as of why. I don't think it's about beta oxidation or something related to energy metabolism. I think maybe it has something more related to your latter point. To be determined. Great question, though. And I'm all ears if there's suggestions.
One more question.
Hi, it's a great talk. I want to know the phase 2 clinical trial. It's either single drug or the combination. And the second question is about the toxicity or the combination. Can you talk about that?
Yes.
Thank you.
Yes. So the clinical trial is single agent in patients who've failed first line treatment. So you know, that's a tough circumstance to look in. This trial it's not supported by industry. It's an academic trial. So I think we can learn something from it, and we're not wed to it giving us these remarkable responses. One of the things we're doing is collecting blood samples from these patients, and we're looking if we're able to inhibit autophagy. Maybe we'll see some outstanding responders, but that's not necessarily the outcome for the trial. I don't know much more than that. You can look on nci.gov to get probably better information than I just gave you. The second bit is in people, the combination has not been tested. Hard stop. In our mouse models, it does not appear that these animals, they don't lose weight, they don't look more sickly. In fact, if anything, they look better because they're not carrying around these tumors that are affecting their ability to digest food and so forth. So there has not been any apparent toxicity. And I also showed you some data at the beginning with the PIK5 knockout, the conditional knockout model in the pancreas. Those also seem to be completely fine. All indications point to that there is going to be a reasonable therapeutic window here.
Thank you so much,
Yeah,
Costas.
yeah, my pleasure.
Right on time. Well done.
Thank you for your attention and questions.